Good afternoon, everyone. Yeah, thank you for having me. I will switch gear a little bit to talk about upcycling of agricultural food waste to animal feed with bioprocessing. I'm also from University of Minnesota. So as we know that a third of our food is actually wasted. It is a global issue. A large portion of the food is actually wasted even before they reach to our dining table. On the other hand, our global population is keep increasing, and 1 out of 9 people in the world are facing a hunger issue, including significant portion of people in America. They also face food security issues. So looking at all this enormous food waste generated globally, so we can, of course, we can probably reduce surplus food production or donate this extra food to hungry people, and down to this ladder, we can throw this food waste to composting or landfill incineration. But what we believe here is we can upcycle this food waste to make them as animal feed and contribute to our livestock industry as novel feed. So this is where we start our research. We expanded our definition for the food waste a little bit, not only including like fruit, vegetable discards, like waste material from food processing, but also include some of the agricultural residues and co-products such as oilseed cake, distillers grains. So we know that the U.S. has more than 200 corn biorefinery process. So all the co-products, distillers grains, and the crop residues. So we have a great potential to utilize all this food waste agricultural residue as a livestock feed. So first of all, they contain enormous of energy and can meet with the animals' amino acid need and the phosphorus need. Specifically for those fiber-enriched materials, if they have more than 20% of fiber, we can divert all these materials as a cow feed. So for instance, like fruit vegetable processing waste, crop residues. And for those protein-enriched food waste or co-products, such as oilseed meal, distillers grains, and some of the fruit vegetable waste materials, so we can utilize them as monogastric animal feeds, such as swine and poultry. So here is a good example for like DDGS. This is the co-products from the corn ethanol industry. So if you look at the DDGS, it has a 5,000 kilocal per kilogram growth energy, almost the same as a corn. It also has a very high concentration of the crude proteins and phosphorus, very ideal for to serve as animal feed. But in the meantime, this approach has a tremendous amount of challenges. If it is a fiber-enriched material, we are always concerned about the content of the lignin. So a lot of materials contains high concentration in soluble fiber, especially high content lignin. So this will negatively affect the feed intake for our dairy cows. And also, a lot of food waste contains high concentration of phytic acid. So this is the main form of the phosphorus stored in the plant materials. And for monogastric animals, like poultry and swine or fish, they don't have a phytase to utilize all this phosphorus. And eventually, this phytate will directly pass through the GI tract, end up in the manure. And then finally, it's a food waste. So in other cases, it contains high concentration of anti-nutritional factors. For instance, canola meal or rapeseed meal, it has a very high concentration of glucosinolate, synapic acid, and this will normally reduce animal intake and reduce animal performance. And in a lot of cases, the poor storage of this agricultural residue and food waste materials will enable the generation of microtoxins. So for instance, like aflatoxin or vomitoxin. So this will make this material unusable. So that's why our group is looking for bioprocessing approach. So this has already been a historical approach. People have been using the fermentation for thousands of years. So if you look at all the pictures here, there's all these delicious food items like blue cheese or soy sauce or kimchi, tempeh. They are all fermented food. So the fermentation, by all means, we're utilizing different type of microbes to go through the fermentation, and then it will change the composition of the food products to make it more nutritious, easily digestible, and more flavor. So so we can utilize this technology to upcycle all this food waste, crop residues, so that they can better serve as animal feed. So if you look at the fermentation industry, so we have a solid state or submerged liquid fermentation. And a lot of cases, we also have to go through some sort of pretreatment process. And depending on what type of cultivation you have, if you have only one strain or two, we can go for the pure culture process, or we can go through this natural fermentation process where we have millions of different bugs working together. And then looking at the fermentation conditions, so a lot of factors will affect the fermentation results. And eventually we can package or dry our fermented feed to provide to the market. So I want to share a couple case studies. The first one is the corn ethanol co-product. So as this research is funded by the National Science Foundation, and we just finished this project last year. So as you know, we have 200 corn ethanol refineries in the US, and literally converting 50% of corn generated in the US into ethanol. If you drive cars, so you know that every gallon of gasoline you consume in the car, 10%, at least 10% comes from ethanol. So this is an enormous industry in the US, and making the US as a global supplier for the animal feed. The reason is after the ethanol is distilled, we dry everything into a co-product we call the DDGS. And this is primarily serving as a cattle feed. And then right now we are seeing more and more aggressive application in the swine and poultry and even like a fish industry. So the reason why the DDGS is primarily utilized in the dairy and cattle feed, because it has, even though it has a very high concentration of energy, a high concentration of crude proteins and phosphorus, but the proteins, the amino acids do not have enough essential amino acids, and the most phosphorus there are in the form of the phytate. So this is where we want to start our fermentation. When we look at the corn ethanol refinery process, so basically we have 3 steps, and each step has many issues. So the first step is trying to grind the corn and go through the fermentation. So we need to add a lot of water. This is a very water-intensive process. And then during the fermentation, we're converting 70% of the corn material, which is a starch, to ethanol. So by doing this, we're literally converting human food into fuel. So we are always have this kind of ethical debate between fuel and food and the fuel. So we are literally removing a lot of human food to supply our fuel market. And the final step, after the ethanol is distilled from the corn ethanol process, we have to go through a very intensive process to dry all this whole sludge into DDGS. the DDGS, the water content of the DDGS has to be within 20% in order to have a better preservation. So this is a very energy intensive process. A lot of natural gas need to be burned to provide a drying power for this whole process. So in order to minimize all these environmental, water, and energy issues for this process, what we have been doing. So we are utilizing all the low quality agricultural residue materials combined with the whole silage and do a bioprocessing, do a fungal fermentation. By doing this, we will be able to double or triple the feed production from a corn ethanol biorefinery. So, because we consume 70% of starch, remove 70% starch to the fuel market. If we combine this low quality agricultural residue materials here, we will triple our feed production. So we can make up all this lost material for the feed market. And we will also enrich our protein content, make the overall products more digestible, enrich essential amino acids. And all these results, and also reduce the microtoxins. So these results, we already published a couple of papers. They are listed in this right corner. If you're interested, you are welcome to have a read. So by doing this, we will be able to triple the animal feed production for our monogastric animals. We will also make those feed more nutritious, more easily to be digested. And overall, we will be able to reduce the environmental sustainability impact of the corn ethanol industry. The second project I want to share with everybody is about the oil seeds, byproducts from a cover crop oil seeds byproduct. So if you live in the northern part of the United States, you know that especially like Minnesota, we have a half year, the soil is not covered. So the farmers are encouraged to plant cover crop. So especially we want to promote farmer to grow the oilseed cover crops such as pennycress, camelina, sometimes even like canola. So the reason is they can harvest the oil seeds. The oil seeds can go through the like a regular oil seeds processing to produce oil. So by planting the oil seeds cover crop, the farmers can make this cover crop as a sort of like a cash crop. So this will provide essential economic incentive for farmers to adopt the cover crop. But when we look at this oil seeds, oil is a main product. This will be the oil similar as our cooking oils, right? Vegetable oil. We can use it as a cooking or for cooking, and we can also use for biodiesel production. But in the meantime, we're going to generate oil seeds meal. This oil seeds meal, especially from pennycress or camelina and canola, they have a very high concentration of lignin. They have a very high concentration of anti-nutritional factors such as glucosinolate and synapic acid. So that's why we propose this solid state fermentation. The primary goal is to reduce the glucosinolate so that we can have a much higher feed intake for monogastric animals. So the overall, by having a better feed application for the oilseed meal, we will have a better economic return for farmers to plant the cover crop. This research we have already generated a couple of papers, and these papers are listed on the left corner of this presentation. The final project I want to share with everybody is this sequential fermentation for novel cattle feed. So this is in collaboration with a research group at the University of Pennsylvania, and this research is funded by the USDA. So we are primarily targeting at fruit vegetable waste as well as like crop residue like wheat straw. So we design this, if we are targeting on the cattle feed, so the primary goal is to see whether we can reduce the lignin content and also provide a better preservation method because the food waste is very easy to spoil. So we designed this a sequential fermentation process. So the first step is going through a fungal aerobic fermentation process. We inoculate the white rot fungi into this waste agricultural residue materials to reduce the lignin content and also break down the lignocellulosic structure. And then we go through the anaerobic fermentation process. This will be very similar to the ensilage process. So we will use lactic acid bacteria, a natural fermentation to drive down the pH, mainly to produce lactic acid. So we can reach the goal that the pH is dropping to around 4. So the final product here will have a much higher digestible fiber, higher quality of the proteins, and very stable pH that will be increasing, like palatability, also can preserve this feed for a longer period. So by doing this, we can improve the overall sustainability by providing the upcycle value for food waste and agricultural residues. So we are starting to publish a couple of papers from this research, and these are listed on the upper corner of this slide. So currently, we are working on a scale up. Some of the process actually are already available in our agricultural community in the US, such as like a silage. Some of the process are relatively new here. So, but we can take references from other traditional food generations, such as soy sauce production or mushroom production, to scale up this process. So basically, we want to look at how to provide better mixing, how to provide a better temperature control in the rural settings, and if it is an aerobic fermentation, how to provide the air and have a better fermentation design so that it can be widely applied in rural America. Of course, for this research, we need to in order to push this technology to the market, we need to have some regulatory support and also a market need for this fermented feed. So that's all for my presentation. I want to acknowledge my students and collaborators, both in Minnesota and Pennsylvania. And as I mentioned, this research has been funded by the Forever Green Initiative in Minnesota, USDA, and the National Science Foundation.